Hi, Miss Nikki here. Welcome to Chapter 23. We're going to talk about patterns of gene inheritance. So Mendel, dominant, recessive, phenotype, genotype, Punnett squares, pedigrees, all that fun stuff. If we're going to talk about inheritance, the first person we need to talk about is Gregory Mendel. He was a monk and a mathematician, and he decided that he was going to investigate inheritance patterns in plants in the 1800s. So he was trying to figure out what's this factor that's being transmitted to offspring. We now call these genes, and they're found on chromosomes, but Gregory Mendel was kind of the father of genetics. So he really is the first person to talk about these packets of information that are being transferred or passed on through generations. Some terminology we need to review: homologous chromosomes. So these are homologous chromosomes. This one was inherited from mom, and this one was inherited from dad. They have alleles at specific locations. So we're going to say this location will denote hair color. And mom has a big G, we're going to say that's brown hair, and dad has a little g, let's say that's red hair. The other term here is sister chromatids of duplicated chromosomes. So these are sister chromatids. Remember that the DNA has to copy itself before we can split and form new cells. So we can see these are sister chromatids. These are also sister chromatids. Even though Mendel had no knowledge of chromosomes, he came up with the law of segregation based on observations. So he looked at these pea plants and he said they must have two factors for each trait, and one of those can be dominant. So one trait could be hidden. So remember our homologous chromosomes, and we have alleles. That would be the factors. So he's saying each pea plant has two factors for each trait. We'll say this is, I don't know, seed color. The factors are going to separate when we form gametes. So remember meiosis, we're going to separate, this is diploid. During meiosis, we're going to become haploid, right? Each gamete contains only one factor. Check. Fertilization gives new individuals two factors. So if I take this egg and then I have a sperm, if we're talking about humans, I'm now going to have the sperm fertilize the egg, and I'm going to end up with two factors for each trait. We have the term phenotype and genotype. So phenotype is going to be the individual's appearance. I think of physical appearance. Genotype is going to be genetic. What do the chromosomes look like? What alleles are the chromosomes carrying? So GG, PP. Here's a nice chart for us to look at genotype versus phenotype. So genotype can be written in letters. It can also be written in words. So here we use the uppercase letter to denote dominant, and we use the lowercase letters to denote recessive. So you can see here, same dominant. That means both alleles are dominant. Heterozygous means, right, different, opposite. So we have one dominant, one recessive, and homozygous, same recessive. You have two recessive. So what does this genotype mean for phenotype, what you're going to look like? So if you have one chromosome with a big F and another chromosome with a big F, remember these are homologous chromosomes. So if you have both alleles are dominant, you're going to end up with freckles. It's going to physically display as freckles. And remember, these are alleles that are coding for a specific protein, which is then going to have some sort of function within the body. So some cases you just need one dominant allele in order to exhibit the dominant phenotype. And then if you have the recessive alleles, then you'll have no freckles. If we're going to look at inheritance and we wanted to look at kind of the simplest experiment, we could do a one-trait cross. We would use this to determine what's the genotype of the parent, what are the possible gametes, combining the gametes, what kind of genotype and phenotype would the offspring have. This example is going back to the freckles and no freckles that we looked at. Here's a one-trait cross, and we do know, so this is known parent genotype. I'll show you a test cross in just a little bit if you don't know what the parents are, but here's just the basic example. So we know that both of the parents are heterozygous, right? They have one dominant and they have one recessive allele. We know if these are the parents, what's going to happen to these two traits? They're going to be separated during the creation of gametes. So here are the eggs, here are the sperm. What are the possibilities? So this parent is the male. The male can give big F or little f, right? If we split these down the middle, big F or little f, that's all they can give. And then for the female, what can she donate to her eggs? Big F, little f. And the way that the Punnett square works, these traits go down and these traits go across. So here we have big F, big F. This one goes down, big F, little f. Little f, big F, little f, little f. So what we're saying here when we look at the Punnett square, these are possible offspring. And we can see that 3 of them, 3 out of 4, are going to have freckles. And 1 out of 4 is going to have no freckles. Here we have a one-trait test cross. So sometimes the parent genotype is unknown. So we know that the male has freckles, but we don't know if the male is homozygous dominant or if it's heterozygous. So we can look at the offspring and we can make a guesstimation as to what is the genotype of the unknown parent. So usually you want to do a test cross with a recessive, because we know that the female can only donate a little f. That's all she has to give. So now look at the offspring. If I mated these two, I know this one is no freckles, right? I know the mom, it carries 2 genes for no freckles. And I look at all of the offspring. What's the phenotype? Phenotype of all the offspring, they all have freckles. If mom can only donate the little f, where did the other dominant allele come from? It had to have come from the parent, and we know the parent has freckles. In this case, there are no there are no offspring with no freckles. So what's the likelihood of the genotype of the parent? The male parent is probably homozygous dominant. Now we look at this scenario, and we can see mom still can only donate little f, right? The non-freckled allele. But here we have 2 offspring that show up and they have freckles. So we know that one of the dominant traits had to come from dad. And then here we have 2 recessive, homozygous recessive offspring. Where did this other recessive allele have to come from? It had to come from dad. So we know that in this case, in B scenario, the genotype of the parent, of the father, had to be heterozygous. There is some statistics and some math involved here, probabilities. Let's just walk through information and how it could be worded. So here, if I said take a homozygous dominant male and mate it with a homozygous recessive female, we know that the female can only donate the allele little f, and the male can only donate the allele dominant allele big F. So tell me something about the offspring. So what is the phenotype of the offspring? 4 out of 4 or 100% have freckles. What about the genotype? What can you tell me about the offspring? 4 out of 4, 100% are heterozygous or big F, little f. For this example, if I said take a heterozygous male and mate it with a homozygous recessive female, tell me about the offspring. So what do we know in terms of phenotype? We have 2 out of 4, which are red, so they have freckles. And we have 2 out of 4, which have no freckles. So this would be 50% have freckles and 50% don't have freckles. So sometimes they'll put this in ratio form. So we're saying it's a 1-to-1. In terms of genotype, what do they look like at the genetic level, at the allele level? Well, we're going to have 2 out of 4, which are heterozygous, and 2 out of 4, which are homozygous recessive. Again, this would be 50% heterozygous, 50% homozygous. If we look at ratio, this would be 1-to-1. When we have 2 traits, obviously this becomes more complex. So here's one pair of homologs, and they're carrying the freckles or no freckles. And then we have these shorter chromosomes, and they're carrying alleles for short or long fingers. And remember, from meiosis, we're going from a diploid to haploid. So we're splitting these homologs up. Now, you can see that there's a random or independent assortment. We don't know exactly which chromosomes and in which order they're going to line up on the metaphase plate, either in meiosis 1 or meiosis 2. So let's look at the gametes here. Here we have big S, right, the dominant allele, so that's short fingers, and then they have the dominant allele for freckles. So if this gamete gets fertilized, you're going to have a child with short fingers and freckles. Same here. What about the next set of gametes? This is going to be long fingers and no freckles. This gamete's going to be short fingers and no freckles. And this one is going to have long with freckles, right? So this is how we get kind of this independent assortment of chromosomes in the different gametes. We also have to think of the fact that we don't know which of these gametes is going to be fertilized. Maybe this is the gamete, in terms of humans, that the sperm will penetrate and turn into an embryo and then a fetus and then be born. So we don't really know which alleles are going to make it into the gamete and eventually into the fertilized egg or zygote. A dihybrid cross is a little bit more complicated. You can see the Punnett square here. There's several more squares. We're taking 2 homozygous parents. So this is homozygous recessive for both traits. And this is homozygous dominant for both traits. And we're mating those individuals, and we'll end up with this first generation where everyone is heterozygous. So remember, the only alleles that this parent can donate to its gametes are the recessive alleles. And this one, the only gametes they can donate are the dominant alleles. But then when we have these heterozygous individuals and we mate them together, we're going to come up with several different gametes. So you can have these 2 alleles, or these 2, or these 2, and these 2. So here we have big F, big S, big F, little s. Then we have little f, big S, little f, little s. So when we mate these individuals, these are all of the possible offspring. And you can see they've kind of added everything up for you. If you want to go by the color and count them, feel free. But there are 9 offspring with freckles and short fingers, 3 with freckles and long fingers, 3 with no freckles and short fingers, and 1 with no freckles and long fingers. So this is a 9 to 3 to 3 to 1 ratio. So how do we use this information from test crosses, from following Mendel's laws? How do we use this information to look at the probability of offspring having a specific trait, right? This might be helpful if we're looking at genetic disorders. If parents have recessive alleles, we might want to know the percentage or the probability of passing that allele on to their offspring. And we can do this also looking at pedigree analysis. I'll show you on the next slide. If we do have a trait that's controlled by a single allele, so we just saw single allele, right? If the trait is caused by a single allele on any chromosome 1 through 22, that's our autosomal chromosomes. We do have some genetic disorders that happen on the sex chromosomes, which are X and Y, which this would be our 23rd pair of chromosomes. If we wanted to trace back specific alleles through our family ancestry, this is where we're going to look at pedigree charts. So the circles are females and the squares are males. And here in this generation, in the 4th generation, we have 2 out of 3 individuals that are affected by a recessive disorder. So we want to go back through the family and see which family members were carriers, maybe which ones still are carriers, so that we can know how this is going to affect future children, right? So by looking at the genotype of the affected, we can kind of go back and try to figure out what some of these previous generations had to look like genotypically in order for this generation to occur. There are several autosomal recessive disorders that are listed in the textbook. I just wanted to mention this one, PKU. This is one of the ones that we do newborn screening on. So when the baby's first born and we do a little heel prick and we put a drop of blood on a slide, one of the things we're looking for is whether this individual lacks the enzyme necessary for normal metabolism to break down phenylalanine. This is one of our amino acids that we need to make proteins. So they're lacking the enzyme that breaks it down, so the phenylalanine builds up. And so all we have to do is put you on a low phenylalanine diet. And if we don't do this, if we don't catch this newborn child, they consume a diet with phenylalanine in it, it causes intellectual disabilities. So they become mentally disabled, and this is something we could fix very clearly early in life if we just limit the amount of phenylalanine in their diet. Another autosomal recessive disorder is sickle cell disease. So again, I keep saying recessive. You can have homozygous dominant. This is normal. You can be a carrier, so you can be heterozygous, or you can have sickle cell disease and be homozygous recessive. What happens, these individuals make abnormal hemoglobin. Hemoglobin is a protein. So they're lacking the gene to make this specific normal hemoglobin. Theirs is a sickled shape. And so this sickled shaped red blood cell is going to clog blood vessels. It's going to cause the breakdown of red blood cells. It can cause internal bleeding or hemorrhaging, poor circulation. So there's lots of issues. This is another one that we test for with newborn screening in, I believe, every state in the United States. If we move on and look at a pedigree of autosomal dominant, you'll see that this looks much different. There's more yellow, right? There's more affected than if we had recessive. So on the recessive, the only way you have the disorder is if you have both recessive traits. With autosomal dominant, you are affected if you have either one of these genotypes. So with autosomal dominant, you only need to have one allele in order to have the disorder. Marfan Syndrome is one of those. It's caused by a defect in elastic connective tissue, which is everywhere in the body. So it's a protein that is not working. It's defective. And this can cause dislocation, longer limbs, longer fingers, kind of a caved-in chest. It can disrupt the aorta wall, which is your major blood vessel leading from the heart that's carrying oxygenated blood. It can increase your risk for an aneurysm or stretching or even bursting of the aorta. Since Mendel, we've learned that some genetic patterns do not follow the strict dominant and recessive. This is incomplete dominance. That means that the dominant does not completely mask the recessive. So they're making this a little more confusing than it needs to be. So let's do big H, big H, big H, little h, little h, little h. What they're saying is the homozygous dominant is going to have straight hair. The homozygous recessive is going to have curly hair. So here we have our heterozygous. Normally, in normal dominance, this individual would have straight hair as well. But with incomplete dominance, this dominant trait cannot mask this recessive trait enough, so you have an individual that has it's kind of in between straight and curly hair. So the heterozygous has its own phenotype. Another deviation from straight dominant and recessive is both codominance and then also this multi-allelic example, which is blood. So if you're type A or B or O, that would be multi-allelic. That means you have several different possible alleles. So there's 3 alleles here that control blood type. That's multi-allelic. If you get these two, if you get A and B, they will actually make you codominant. So you're expressing both. Remember, these are genes. These are variants, which are alleles, and they code for proteins. So if you have A allele, you're going to have a red blood cell that has A tags on it. If you have the B allele, you're going to have B tags. If you have O, I call them naked. They don't have any tags. But there are some individuals where they will get both the A allele and the B allele. So you got this one from mom, and you got this allele from dad. So you end up with AB blood. You actually have both tags on your red blood cells. So the AB would be an example of codominance, and the ABO would be an example of multi-allelic. Here's a nice example of the inheritance of blood types. Here we have an allele for B tags, and the lowercase I means we have an allele for O tags or no tags, right? Here, this individual, this mother, has one allele for A tags and one allele for O tags. We can see that mom can donate either one of these alleles and dad can donate either one of these alleles. So you could end up with an offspring that has AB blood. You can end up with an offspring that has BO blood, one that has AO blood, or one that has OO. So if you have a mom that has B blood and a dad that has A blood, you can actually end up with a child that has blood type O, because they were both hiding these recessive O blood type alleles. You might also hear people use this term Rh factor. So you might hear somebody say, I'm blood type B positive, or I'm B negative. So a positive person is going to express this Rh antigen on the red blood cell. So if I'm B positive, here's my red blood cell. I'm going to have B tags, and then I'm going to have these Rh positive tags. If I'm B negative, I'm going to have the B tags, but I'm not going to have any RH tags. And this becomes important during pregnancy. If these RH factors do not match between the mother and the child, it can cause some issues. Another inheritance pattern that's a little different than dominant and recessive is called polygenic. So there are several genes responsible for creating skin color and the various arrays of skin color that we have. So basically, I like to think of it as the more dominance you have, the more dominant alleles, the darker skin color. So more dominant alleles, it kind of has an additive effect. So you can see people that have the darkest skin have more dominant alleles, and people that have lighter skin have more recessive alleles. There are also environmental influences. So we're now starting to see that genetics and environment play together for optimal expression of a trait. So what does all that mean? If you have a polygenic trait, something like height or skin color, it can be influenced by the environment. So think about height for a second. Height is also dependent on your diet. Does it matter? Nutrition, the food that you consume. So remember, the food that we consume provides the amino acids that we need to make proteins so that we can grow. So it's your genetics, but it's also your environment. And we see this with flower colors. If you know anything about like hydrangeas, depending on the soil pH will depend on whether the flowers are more purple in color or whether they're more blue in color. So that's an environmental factor affecting the genetics of the flower. We also see this in rabbit fur, specifically whether we have more white pigment or whether we have this melanin and produce a darker pigment. So there are some environmental influences acting on your genetics. Well, that finishes up chapter 23 in gene inheritance. Make sure you go back and look at all of those terms. Sometimes we think we know the term and then we get to a test question and the way that it's worded kind of throws us off. So probably the best thing to do is create flashcards and make sure that you know all of those terms. What's homozygous recessive, homozygous dominant, what's phenotype, what's genotype, and then list out all of the different types of inheritance. What's autosomal recessive, autosomal dominant, incomplete dominance, codominance, right? So I'll talk to you guys next time. Bye.